Thank you all for joining. I'm pretty excited for today's webinar. So this is the third in a row of our season of webinars that we started last year. And this one is quite special because it's more focused on protein technologies. So today we will tackle a couple of things that we developed at Acies, but also talk about a little bit about also where the proteins and peptide technologies are currently. And basically the agenda is quite similar to the other sessions that we have. We will have a quick Acies Bio introduction. Then I'll tell a couple of words of state of the art of the protein peptide technologies. And then we will talk about our platform. So what we developed in terms of our Bacillus subtilis host, what we did in terms of achieving high titers. So the showcase that we now also made publicly public. And then we will also talk about our protein peptide production platform, which we now developed, and we will launch it with this month. And today with me, I have three speakers from Acies. So Martin Kauszczek will tell a little bit about Acies. So he's heading the business development, so he really knows Acies from inside out. And with us, we have also two of our experts, Vasilka Magdelska, who is leading our synbio unit and is also the pioneering researcher of our protein platform, and Anna Maria Staffa, who is head of protein platform and will also explain a little bit about our peptide platform. So I think let's start with the Acies Bio introduction. So Martin, please take it away.
Okay, hi, also from my side. Really happy to see so many people joining today. I'm not gonna talk too much about Acies Bio in general, but just to give you the outline how we can work on different technologies and how we can then plug in the protein technology to all of the other technologies that we are developing. So essentially, Acies Bio, what we do is a streamlined development process, microbial process, where we can start with really early idea generation and finish with a contract manufacturing, basically solving all the needs that industrial biotechnology needs in order to in order to come to the market. And we are talking about strain design, then optimization and development of microbial production strains, and then slowly going up in the TRL curve towards developing the bioprocess, developing the downstream processing, and then do the commercial demonstration and CMO that we do at our commercial demonstration unit that we are expanding this year from 10,000 liters fermentation that we have currently to 30,000 liters fermentation that will be installed later this year. At Acies Bio, we've done that for the last 20 years. And currently we are a bit more than 100 research scientists, worked on more than or around 150 projects in diverse industries and diverse TRL settings, from discovery projects to enabling the technologies to come to the pilot scale, and very importantly also made projects that reached industrial scale production on the different scales, from really huge projects and products to smaller, more niche production projects. We've done that development for diverse industry segments, such as specialty ingredients for food feed, pharmaceuticals or nutraceuticals, industrial chemicals, agriculture, and because of that maturation of the projects, we can use tailored business models for each of the industry segments and let's say, technology readiness of the projects that we are onboarding with our partners. So that's gonna be all from the general Acies Bio presentation, and I'm giving my the slides now to Bor to introduce us more specifically in the recombinant protein technologies.
Thank you, Martin, for this introduction. So in the next couple of slides, I will speak a little bit more about recombinant protein technologies, where they came from, and how ACS looks at the relevant industries where they can be applied. So if you look historically, recombinant proteins are mainly quite connected with synthetic biology development. So when the recombinant technologies were developed, also development of protein and enzyme technology started. So it all came together with how the genetic tools were developed, and with early adaptation, we can see that it was implemented in different segments. But let's say the most prominent cases came from pharma, like insulin or monoclonal antibodies. And from there, it grew, let's say, steadily. And then in early 2000s, it really became a synthetic biology boom. A lot of different genetic tools were developed, and a lot of different areas where they were implemented were discovered. And from there, it's then migrated to different kinds of industries, different kinds of technologies. And more recently, we saw that the proteins could be also a source of food. So we had a big investment in times of 2015 to 2021, where a lot of focus was put into precision fermentation, and specifically to developing, recombinant proteins for food. So there was a lot of investment. Let's say the impact was relative, and there was a lot of things that didn't go well, but the major outcome of that was that a lot of investment went to the development of different genetic elements, genetical tools. Different hosts were explored in different kinds of ways. So as we look at now back, we see different major milestone being achieved in that period of time. So, and this is basically the time from where we also, as ACS, started to build our platform and grew in relevance with our Bacillus subtleties. If you look into what kind of proteins the development went, it was mainly, in caseins, egg proteins, food colorants, and then also, if you look, a lot of investment went into lipids that is maybe not that relevant, but also address the food market. So if you look in the recombinant protein market as a whole, it's quite similar to the synthetic biology market. So pharma products are still the main thing that the technology development is focused. So, but that's basically if we look at in the perspective of revenue. But if you look in the share volumes of the enzymes or proteins produced, then food and feed segment and also agricultural space became much more relevant. So those established markets are now growing steadily, and synthetic biology and recombinant proteins are here to stay. How we implement them are mainly dependent on the how the technology, what cost can the technology take, and how long the development takes to really get the product to the market. So at ACS, we see all those segments quite relevant, but maybe to highlight two of those that are more prominent in recent years is agricultural sector, where we also as a company focus quite a lot. So agricultural sector is an emerging sector of biological solutions. If you look back, a lot of focus was done on biosolutions that were based on live microbes that often lacked efficacy and specificity. And if you look into conventional pesticides, the overuse just built up so much of the resistance in the pathogens that the pesticides just don't work. So alternative solution is demanded, and it has to be implemented soon. And if you look into where companies are focusing, so proteins and peptides really offer a controlled, targeted pathogen disabling. So you can really approach some physiological interactions that can really disable a specific pathogen and be very effective at small quantities. Also, a good aspect of the agricultural solutions is that many different kinds of technologies can be applied, and also hosts are for some proteins more relevant than for the others. And for peptides, you can achieve a lot of effect with the small concentrations of the specific peptide. So, and if you look into agricultural sector as how the technologies are developed, it's really important to be cost effective. And if you look into the conventional production of proteins and peptides, which originated from pharma, we see that the costs of such technologies are really too high, and that the resources are too high to really implement on large scale, agricultural sectors that are now being explored. So fermentation-based technology really gives an opportunity to enable such technologies. The second sector is the one that we all really know. It's AI, and it's not really, I would say, it's not only relevant in our industry, but it's omnipresent nowadays. But anyhow, what we saw from our experience and from the companies that we work with, protein engineering is really one of key applications that AI has high relevance. So developing new enzymes, developing new proteins with new abilities, or even de novo design now became really fast on a fast track, and it can be really enable technologies that previously weren't even possible to think about. So, but how these companies work and how we saw the collaborations that we were in, it's really different. So you have companies that really focus on specific segment of the proteins, let's say food proteins, finding, screening for new proteins that can be then scaled. And then you have like software platforms that really enable optimization of different kinds of proteins at once. And you are really the one that guides the development, and one can focus on your specific targets that you want to achieve. So this sector is, I think, really will stay and guide the further development technology, but it's also important to raise it, awareness about it, because it also depends a lot how then these technologies that are engineered by AI models will be then also implemented in a real industrial environment. So building the production technology is also really relevant and has to be co-developed together with the protein engineering parts. So, and with this slide, I would conclude the overview of the protein technologies, and maybe give the word to our experts, that they will, Vasilka and Anna Maria, who will tell a little bit more how we developed our platform to be really useful for different kinds of proteins, and how what we see in terms of technology is relevant to develop and focus on to achieve high titers. So Vasilka, I would like you to join the discussion and take over the presentation.
Thank you, Bor. I will take the control now. Hi to all. I will present the protein platform, what we developed in Acies Bio. First, I will present the protein platform as a general, then we can go and more focus to introduce also our Bacillus protein platform. So our platform, basically, we started with developing 5 years ago. We have a broad range of prokaryotic and eukaryotic expression hosts, and the choice which host will go in further production and to be taken for a particular protein production, it depends also from the nature of the protein, and we are deciding that at the beginning of the project. This allows us very short timelines for executing the experiments and also high flexibility. So during all these years, we have been working with many different proteins, even with complex ones. So what is the specificity of Acies Bio protein platform? Our main advantage is a very short and fast proof of concept phase. So this is the initial phase when we are confirming that particular host is very optimal for production of the target protein. We are starting with several steps. The first step is in silico prediction, sequence optimization, and also making some execution plan to be fastly successful. So in the next step, it's starting with laboratory work, making the constructs and generation the strains, production strains. And after the fermentations, we can analyze the titers, potential, and secretion capabilities. So when the strains are at this stage confirmed, we are going further on in the next step with bioreactor production. This is the bench scale bioreactors. We are fermenting in defined media, and at this stage, the production mainly we are achieving up to 15 grams per liter of the target protein. So when this is the proof of concept phase, and when it's finished and we have successful results, then we are going in the next phase. This is the optimization phase when we are improving even further the protein production. At this stage, we are starting with strain optimization, genetic elements optimization. Then we can go also with random mutagenesis and high throughput screening if needed. Then we have a fine-tuned media optimization and bioprocess optimization together with feeding regimes. At this stage, as we reported previously, the maximum levels of the protein production we achieved at Acies Bio is 45 grams per liter. So I think this is the most important stage when we are reaching the highest production levels, and with this success, then we are going in the final stage, industrialization phase, where we are scaling up from bench scale. We are going in the pilot scale. Acies Bio capabilities are from 100 liters up to 10 cubic meters of fermenters. Additionally, we have also DSP, very strong DSP department with extended knowledge of the protein purification. So in the DSP department, we can develop the DSP process from the scratch, and we are taking in consider the customer's requirements for the purity of the protein and also for the final cost of protein production. So this is our protein platform in general. Now we can go further on with the Bacillus subtilis protein platform, and we will show our showcase with high titer protein production. So in the slide it's further. So the Bacillus subtilis, Acies Bio Bacillus subtilis platform is built from strains, engineered different background strains. All of them are non-sporulating. They are protease deficient, have high secretion capacity, and some of them also they have some additional genetic modifications for improving the transcription and translation. We are using strong promoters for expression, different expression vectors, and also signal sequence library to select the signal sequence which has a high efficiency for production and also for secretion of the target protein. What is very important for our protein platform for Bacillus, that we have both capabilities to express from plasmid-based, to have plasmid-based expression, and also stable genomic integration without presence of any antibiotic selection marker. So in parallel, we developed also defined low-cost production media, which is allowing us very stable and scalable protein production. And besides the strain engineering, also bioprocess development, it's very important for high protein production. Here we use a streamlined seed train and standardized inoculum strategy. The inoculum is prepared in two stages, and our defined media, together with controlled feeding regimes, is allowing us a very robust, reproducible, and stable protein production. This is on this slide is present our workflow for proof of concept phase with the Bacillus subtilis protein platform. How we are starting? We are starting with screening of signal peptides under the strong promoter, and after the preparation of the constructs, we are transforming our host strain, and then after the selection and confirmation of the transformants, we are ready for screening of the different strains. And following this, it's analysis of the results, and the best strains achieving the highest production, we are retesting further on in shake flask fermentation for confirmation of the strains. At this stage, also we are shipping to our customers also the material for further analysis. So on this slide, we are summarized our smart route to protein using Bacillus subtilis host organism. So we started at Acies Bio, as I mentioned, several years ago with scale of milligrams production, and with several rounds of iterations, we were able to significantly improve the protein production, even 40-fold. So how we achieved this? We started also with many different approaches. So we developed additional background strains, modifications of the strains, then optimization of genetic elements, bioprocess optimizations together with feeding regimes. And we were able to achieve this high level of protein production. So in all these years, we have been working with many different proteins, as nanobodies, very different enzymes, agroproteins, food and feed proteins, and we were able to produce these proteins from several grams per liter up to 45 grams per liter production. So at this point, in the last year, we have developed also a peptide platform based on our protein platform, and in which we achieved very good results, very promising results. At this point, I will leave my word to my colleague Anna Maria, and she will present the peptide platform, and she will lead to our success more in detail. So Anna Maria.
Thank you, Vasilka. So the recent addition to our Bacillus subtilis protein platform is the peptide platform using the same host, Bacillus subtilis. So we actually offer quick proof of concept phase. That is the evaluation phase where we are checking the potential for the titer of a specific peptide. We check whether it's going to be stable, and we optimize purification. So in this evaluation phase, we actually have really fast preparation of samples for in vivo trials for testing their activity. Further development in scaling up phase allows the development of industrial media, bioprocess setup, and USP and DSP that we have in-house. And then this complete development actually allows more than 50 grams per liter titers. And with further development, we actually can go to industrialization. So the main difference between Bacillus subtilis peptide platform and Bacillus subtilis protein platform is in the first phase, in the proof of concept. So here we have developed a library of carrier peptides and carrier proteins. So when we are onboarding a specific peptide, we are making fusion constructs for the expression with different peptides and plasmids peptides and proteins from the library. And we are transforming Bacillus subtilis with the plasmids used for expression. After the transformants are collected, we are screening them for in fermentation for the production of a fusion protein, and then we are analyzing the protein expression by SDS-PAGE and Western blots. So those transformers that are top producing, they are retested in shape flask, and then after retest, the supernatants are collected and peptide is cleaved from the fusion protein, purified and prepared for the shipping. So as I've mentioned previously, we are making fusion constructs of the peptide with a specific carrier protein or carrier peptide. And here you can see on the gel how actually this pure cleavage and purification looks like. So the fusion protein is shown here in the lane marked with B, and then the target peptide is cleaved. And here you can see two bands of the target peptide and the carrier peptide. And after cleavage, the target peptide is purified either by precipitation or by different chromatographic methods. So we are reaching up to 20 milligrams of a target peptide in initial first proof of concept work package, and then with scaling up, we are reaching final titers of grams per liter at benchtop bioreactors, and then in the big bioreactors, we are ready for industrialization with technology development in tens of grams per liter.
Thank you, Anna Maria, and thank you, Vasilka, for this nice introduction into both of our platforms. So the idea today was to really explain the development through our platforms, how we see it. It was like a fast presentation of our capabilities, but also showcasing where the key steps are in the technology development. So quite clearly, one of those is strain development, which takes time. As we said, our platform now is quite old, and the strains have evolved through years. I experienced it. I experienced that myself in the lab and also as in the business development. So I would say that before I go to Q&A, that there are some key takeaways that we can take from these presentations. So the application really defines how you will develop the protein technology, protein production technology. So if the enzyme has unique post-translational modifications, it's also quite clear that maybe sometimes Bacillus is not the relevant host. You have to always think about also what your end goal price is. So a lot of times, people start historically from E. coli, and then they make the calculations and come to conclusion that basically the DSP is so expensive because it's all the protein is intracellular that they have to reinvent the whole technology. So that's why we also focus on Bacillus subtilis. So it's a secretion host. It secretes the protein intact and also active. So it really presents a unique proposition in terms of DSP development. And it's also what I would like to point out, always think about USP and DSP development together with the development of initial innovation that you are thinking about. So we emphasize that also in our previous webinars and try to advise also to our partners. So and why we develop the platform. So platform really helps you to validate the different technologies quite quickly. So proteins can be tested out with robust genetic tools. That's not only for Bacillus, but also for other hosts. So we think that platform is really for early conceptualization really needed, but for longer development, then you have to also have this capabilities and knowledge to really specifically develop the strains. So, and in terms of Acies, what we do, and we now quite extensively showcase that, so we can really guide you through all those steps of the development. Specifically for Bacillus, we already have, set up genetic tools, set up bioprocesses, and also everything scaled at our pilot scale. So this really becomes in handy as soon as you develop the protein technology. So, and basically, I would like to conclude this presentation with inviting you to our Q&A session. We have an open Q&A where you can post the questions. We intentionally make the presentation a little bit shorter to give room for all the questions you have in the Q&A. So we will go answering those in periodic order as you stated them. So please go ahead and start with questions, and I will try to guide through all the questions that we have. So maybe we can start with, yeah. But Philip raised also a question in the meeting chat. We do in silico prediction within the platform integration of our workflow. So once we get the sequence, we analyze what kind of enzyme or protein it is, how we can that successfully translate to our strains. So what we have in-house, and maybe we didn't raise that quite a lot during our presentation. So we do all the data integration through all our stages of development. So we have digitalized all the data, so expression levels, bioprocess parameters, and try to analyze that and make some empirical decisions down the line. So with proteins, it's always, when you develop the production host, it's always protein dependent. That's why I must state out. But also you can predict to some extent as you get relevant data from different kinds of experiments. So we try to now really integrate that. We have our digital team that really works on different kinds of machine learning models and algorithms that really help us this first in silico development. We also optimize all our gene sequences in-house with our tools, and have seen that it has great potential for those kind of things. So maybe if we go to the Q&A.
Yeah.
So could you briefly clarify the main technical limitations of your peptide production platform that might limit the types of peptides that you can produce? I think maybe Vasilka and Anna Maria.
For the peptides, it's a really specific situation. So we saw that carrier proteins are the best one for screening for the peptides. And that's why we have library of these carrier proteins, because each peptide is not nicely produced with in combination with every carrier. So that's why this is the first step in the peptide production with screening of the carrier proteins. Of course, that some of the peptides, it's really difficult to produce, and this proof of concept phase should be a little bit extended and try some different background strains and different genetic elements. It is not predictable as much as the proteins or especially enzymes, but yes, we have successful even with the difficult ones.
So the next question is, both very small peptides and large structurally complex proteins are known to be a present challenge for secretion-based expression system. Maybe you can read the whole question because it's really long in the Q&A. Yeah.
I think we don't.
So we maybe to start, and it's true. Small peptides are problematic, and big proteins are problematic to secrete. That's quite logical, I would say. Even so, let's say for peptides, you always have, the calculation between doing that in a solid phase synthesis or doing that via fermentation. So there are two things. So one is what will be the end application. So the cost will be the main driver here. And also the size, at what size this solid phase synthesis is just not efficient anymore. So we all know that those syntheses lack efficiency as you go in bigger peptides. So that are two things. So one aspect also already that Vasilka mentioned is carrier proteins. So that is something that we can maybe skip and also bypass some of the things. And then if you go in the queue.
Yeah, let's answer this one regarding the protein. Because it's quite a lot of questions. Some of them we might get in contact with you because maybe we don't have time for all of them. But we can comment on what protein we achieved 45 grams per liter approximately, and if it was repeatable robust and was it genomically integrated construct or episomal plasmid?
The production of this showcase that we shown today, it's really robust, reproducible. The size of the protein, it's around 30 kilodaltons. It's eukaryotic protein, which is really difficult for prokaryotic strains mostly, but yeah, Bacillus is producing very nicely. Yeah, I think this is the answer of the question.
Okay, maybe I can also answer some of the IP questions. So, how do we avoid intellectual contamination with the knowledge shared in different projects? So, basically, how we are working on is that the background IP that is on the platform itself, then it is important for production of different enzymes and proteins, stays with Acies, and we are then having the let's say forward IP that is protein specific that we either license or get exclusive partners with a specific partners that we are developing the products for. So that's the way how we try to let's say separate the background and the foreground IP, and of course if there is something that is specific for the successful production of some niche part of the proteins, then we work on that specifically. Maybe also regarding the how much we can, or what's the final prices of the proteins that we can produce using that strains. I think it really depends on the scale where we will be producing, and also the further downstream cost that can sometimes be like really the biggest part of the cost, but sometimes they're smaller. So we are talking about significant reduction of the costs because of the good secretion of the proteins that we are having, and this is giving the highest cost savings, let's say, in the DSP side, and then going towards the higher volumes with our 30 cube meter fermentation, we'll be able to decrease the cost of the protein production by several tens of folds compared to, let's say, what we have now at 6 or up to 10,000 liters. And of course, going beyond this 30 cube meters is giving the further decrease of the cost. Of course, with that we can probably not still go to really final bulk protein prices, but we are achieving, let's say, double digits euros per kilo in a final production process, optimized and scaled to final scales.
Maybe to because there are a lot of questions, maybe to answer a couple of those quickly. I see Florian has a couple of questions, but maybe just to go through yours briefly. So how long does it take specifically to develop a process for client-specific peptides? So I would say the proof of concept phase is 2 months.
Yes.
Yeah, the proof of concept phase is 2 months, and in this phase, we produce the prototype material, see what's the titer that we are achieving, and if we need some further optimization. If we have like really good results, we can directly go into further development in terms of scaling up the technology and already producing the proteins, so peptides. So this is really specific because there was also a question in between I saw on the peptide modifications, which is something that is really can be addressed totally chemically or via enzyme transformations. But those things are quite difficult and really have to be addressed case by case specific, because not only the peptides lengths are different and regions, also the enzymes that can be implemented for those kind of procedures are different. So this is quite specific, but we explored already how to modify peptides as they were produced. So this is something that we are now also quite extensively working on. Which maybe
One question for disulfide bond
Yeah.
formation. Yes,
This-
we have a lot of experience with nanobodies, and yes, Bacillus subtilis surprisingly is capable to manage with these disulfide bondages. We proved that also with non-reducing SDS. We are not in-house testing the activity of each protein that we are producing, but we are leaving this to our customers. That's why at the end of the proof of concept phase, we are transferring also some amount of material to our customers to test the activity. And yes, they were tested in the past and they were mostly active. Yes, more than 90% of our proteins that are produced in Bacillus are active and functional.
Here's a nice question. So do you have automation capabilities enabling testing the expressions of different peptides or proteins at small scale in your different chassis? So we do have, we spoke just, maybe you saw in our smart routes that we have microliter and picoliter scale. So this is basically our microfluidics department where we can really develop those high throughput methods. So and one aspect also in the further technology development is high throughput non-GMO approach to optimization of strains. So there you already have to have the capabilities to really screen for small incremental changes and also to screen thousands and thousands of strains. So that we have in-house, and that is also one of our expertise that we really built throughout the years. So I would say that we have those capabilities, but it depends how you want to really measure the protein or the peptide. Can you tag it? Can you make it fluorescent and so on? So those things are really can be discussed quite extensively. So let's see what we can still. So on the chat side, I will check. Yes, it's extracellular protein. So there was a question. We did it in pilot scale. Our Bacillus was scaled already quite a couple of times. So, and there was a question about minimal media. So what is minimal media? Maybe a few words.
It's fully defined media. I think here was mentioned corn steep liquor, nothing similar with corn steep or corn meal or anything organic in the media. So it's fully defined carbon and nitrogen sources.
And that maybe also answers Michael's questions about the media optimization that we did. So we really went from all the complex components to really mineral defined compositions, because this also enhances the robustness of the process. So it's a main cost driver in the end. So
and easier for purification. DSP, it's much easier.
Yeah. So I think we are nearing the end of the questions. Maybe I missed someone. Maybe we can address that also via email.
Yeah, nevertheless, we'll go through all of the questions and we'll get in touch regarding the specifics, so that you can also ask further questions and.
Yes. So maybe this is the right point to conclude our webinar. Really nice that you made so many, that you had so many ideas and questions about our technology. I hope we give you some insight on how we see protein technologies in general, but also how Acies Bio works in this space, and that our platform also brings some opportunity to you. And we are really eager to see how we can collaborate on different kinds of levels. So thank you all for joining, and I hope you had a great webinar. And this will be also available in a couple of ways on online versions. So Have a nice day and goodbye.
Thank you. Bye. Bye.